Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my session. So hello, hi, a very happy good evening, Vishal. So hi, Prince. Hi, Antonia B. A very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Yes, so tomorrow we are having a mentee and I'm going to announce it. Alright, so today we are in lecture 2 of biomolecules. So I hope you have solved the homework questions. I told you I have 10 questions after the lecture and I told you to solve them. So I believe you have solved, and today we will discuss those 10 questions first and then move forward with the other part of the biomolecules. All right, yes. So today we will finish biomolecules, just the part before enzymes. In the next class we will talk about enzymes. All right, yes, a very happy good evening, Prince. Thank you for joining my session. So without any further delay, so this is the announcement for tomorrow. Yes, Shubham sir, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So tomorrow at 4 p.m. So as promised, I told you I will teach transport in plants conceptually, because when I bring a question about turgid pressure, solute potential, osmotic pressure, it's pretty confusing, right? So I told you I will teach it conceptually. So yes, so tomorrow come join me at 4 o'clock. So I will teach you the concepts of transport in plants. So tomorrow, yes, because we had only one mentee this week, so I thought I will have one more mentee because tomorrow is weekend, right? So 6:30 p.m. guess the chapter, season 1, episode 6. So which chapter? Okay, so I haven't taught, so maybe tomorrow at 4 o'clock if I'm able to give you some clue, so I will give you. Alright, okay Vishal? Fine. So yes, Srinivas, hello. So thank you for joining my session. So please do like, share and subscribe. So this is lecture 2 of biomolecules. So what are we talking about in the previous class? So in the previous class, we spoke about the chemical analysis experiment. So chemical analysis experiment is where you took your living tissue and you mixed it with trichloroacetic acid, okay, and then when you filtered it, so you had two portions. So the two portions were your filtrate, or it is also known as the acid soluble portion, the other one is known as retentate or the acid insoluble portion. So all the living tissues in your living organisms, okay, yes. So this is basically organic, meaning it is carbon containing compounds, okay. So these are known as your biomolecules. So what are the different biomolecules that is present in your body? So that is what we have been doing, okay. I think I guessed I will say tomorrow. Okay, fine. So it is up to you. So as long as you find out the chapter and you perform well, I'm very happy. But even I haven't given a thought as to which chapter I will be taking. Okay, ask me tomorrow at 4. If I have some clues, I can share. Otherwise, directly we'll meet tomorrow at 6:30. Okay. So before that, how did you find about the inorganic components? So about the inorganic components, yes. So we did a destructive experiment. So where you took a living tissue and you dried it, right? And then you burnt the living tissue. So whatever remained is your ash. So this consisted of the inorganic portion, right? So these were the two experiments. So what did the biomolecule portion of your living tissue contain? Yes, Shimpi, hello. Wishing you a very happy good evening. Thank you so much for joining my lecture, Shimpi. So please do like, share and subscribe. Ma'am, from NCERT textbook only? Yes, it will be from NCERT textbook, but maybe the question looks tricky or the options might look close enough. Alright, okay Vishal? Fine. So what are the different biomolecules we started talking about? That is your amino acids. Then we went on to talk about your lipids. Then we spoke about your nucleic acids, right? So these are the three things that we spoke. So about amino acids, what do you remember? Yes, what is the structure of your amino acid? So you have a central carbon, so this is your alpha carbon. So you have H molecule, you have an R group, you have a carboxyl group, and you have an amino group, right? Yes, so this is it. Then you have what is Zwitter ion. So Zwitter ion is when you have neutral charge. So this is at physiological pH. So you have 20 amino acids. So it has been classified as essential ones. That is that has to be supplied through diet, and the other one is non-essential one. Right? So these are the two things. So carbohydrates we have not yet discussed. So only these things we have discussed. So today we will discuss carbohydrates. All right, friends? Okay. Fine. So this is about your amino acids. So then we went on to talk about your lipids. Right? So lipids have two parts. One is your fats and the other one is your glycerol. I'm sorry for the spelling of glycerol. Yes. So this is the two parts of your lipids. Yes, Dharani, a very happy good evening. So thank you so much for joining my class today. Hi Titan, hello. So thank you for joining my class. So please do like, share and subscribe. So this is about it. So in fats, we read about two things. One is saturated, the other one is unsaturated. So saturated is without double bond and unsaturated is with double bond. Yes, so this is the difference between saturated and unsaturated. So mostly your animal fats are saturated. So animal fats include palmitic acid, stearic acid, etc. Then your unsaturated acids or your unsaturated fats include your oleic acid, linoleic acid, linolenic acid, and finally arachidonic acid. So this is about your unsaturated fats. So this I didn't let you know in the previous class. So please make a note of it. You have something known as what is hard fat. So hard fats includes your ghee and butter. So they are basically triglycerides. So please make a note of this. So this I didn't tell you in the previous lecture. So note down what are animal fats. So animal fats are basically unsaturated fats, that is without double bond. So examples include palmitic acid and your stearic acid. Okay? Yes? Fine. So further we went on to talk about your nitrogenous bases. So that is a part of your nucleic acid, right? So we have two types of nitrogenous bases. One is purine. Other one is pyrimidines, right? So purines include adenine and guanine. Pyrimidines include cytosine, thymine and uracil in case of RNA. So you have a sugar. So sugar is connected to the nitrogenous base. So this bond is known as your glycosidic bonds. Now this together is connected to your phosphate molecule. So this is connected via ester bond. So this structure is known as your nucleotide, right? So these are the things that we discussed in previous lecture. Yes, purines have double ring structure, whereas pyrimidines have a single ring structure. Okay? Yes. So moving forward, so I started this but I didn't complete. So what are primary and secondary metabolites? So at the end of so your body, yes, so your body takes in complex molecules, right? So you feed your body food, right? Say for example, we had rice for lunch. Okay, so this rice is broken down. So this is a form of carbohydrate. Why? So because rice contains starch, So which is a form of carbohydrate. So when I further break this down, I have glucose, which is my monosaccharide, right? So ultimately I have glucose. So the process that happens to break down your food molecules into the primary compounds, this is known as your primary metabolites, the form that is utilizable by the body. So this is your primary metabolites. So if you have taken dal, okay, so this is protein, right? So this breaks down into the corresponding amino acids. So this is your primary metabolite. Now, when the products of your primary metabolite are used to produce further pigments, scents, other storage components, that is your secondary metabolites. So metabolites at the end, they are the end products of cellular regulatory process. So this process is must in your body. It happens every day because your rice is not utilizable by the body. That is why it breaks down into a form that is usable by the body, right? So this is your cellular process. The end products of your cellular process is known as your primary metabolites, like your amino acids, sugars, etc. In plants, fungi and microbial cells, you have alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. It is not species, it is spices. Okay, so all these are your secondary metabolites. So when your primary metabolite is used to produce these substances, so these substances are called as secondary metabolites. Now, are you able to get the difference between primary and secondary metabolites? Yes? Okay, so these are the list of secondary metabolites that is given in your textbook. So you should be thorough with this. They might ask you as a match or as a statement type question. So alkaloids are a type of drugs. So remember morphine, codeine, etc. Terpenoids, monoterpene, diterpenes, etc. Essential oil, lemongrass oil or your tree grass oil. Toxins, abrin, ricin. Lectins, concanavalin A. Drugs, vinblastine, curcumin. So this is also, curcumin is present in your turmeric, right? So that is a very important compound. Polymeric substances like your rubber, gum, cellulose, some of these are commercially exploited, but they are still secondary metabolites. Okay, is it clear? Yes? Okay. So shall we go to the homework question? So how many of you did open the PPT and check for the homework question? Yes? How many of you did? Okay, so question 1, ribose sugar is linked with nitrogenous base by O glycosidic bond, N glycosidic bond, ester bond, amide bond. Yes, what is the answer? Question number 1. Yes, so ribose sugar. So this is in case of RNA. So this is linked to the corresponding nitrogen bases like adenine, guanine, cytosine and uracil by what is this bond? So this together forms your nucleoside. Yes, what is the answer? Yes, absolutely right. So this is bonded by N glycosidic bond. Yes, everyone is right. Perfect. Okay. So question number 2. Identify in which one of the following carbon compounds heterocyclic rings can be found. Proteins, amino acids, nitrogen bases, lipids. Yes, question number 2. You should have carbon compounds as well as your heterocyclic rings. Yes, what is the answer? Come on, quick. Yes. What is the answer here? No, proteins do not have heterocyclic rings. All proteins do not have heterocyclic rings, right? So there is the simplest proteins are made up of amino acids. There is one simple amino acid. Instead of R, you have H. So R is replaced by H. This is the simplest amino acid, that is lysine. So it is not protein, so it is not amino acid. Yes, it is your nitrogenous bases. They have heterocyclic rings, that is your purines and your pyrimidines. Right? Okay, so yes, it is option C. So question 3. After doing chemical analysis of organic compounds found in living organism, two fractions were observed, namely acid soluble pool and acid insoluble pool, carbon pool, hydrogen pool, inorganic and organic pool, aqueous and non-aqueous pool. Yes, what is the answer? Question number 3. So when you do the chemical analysis with your trichloroacetic acid, so what are the two fractions that you get? Yes, so you get an acid soluble pool, which is also known as your filtrate, and this is known as your retentate. Yes, so it is option A, right answer. Okay, so let me change the color. Yes, this is the right answer. So next one, question 4. How many of the following are nucleoside? Adenine, guanine, thymine, guanosine, adenosine, cytosine, adenosine triphosphate, uridine, uracil. Yes. Question 4. So nucleoside. What is your nucleoside? So nucleoside is when your nitrogenous bases and your sugar are attached. Yes. 3. Yes, Shimpi. Yeah, okay. Shimpi said B. No. So Prince says A. So A is right. So what are the nucleoside? Guanosine, adenosine, cytosine. Then you have you have only 3. Uridine is uridylic acid. So it is only 3 which are nucleoside. So guanosine comes when you combine guanine with your ribose sugar. So this is known as guanosine. Okay, yes. Then your adenine plus your ribose sugar is your adenosine. Then you have your cytosine plus this is ribose sugar. No, cytosine is not, so it should be uridine, so that is what I was wondering. So uridine is a nucleoside. Yes, cytosine is not, it is just the normal nucleic acid. So uridine is a nucleoside, so it is uracil plus your ribose sugar. So this is uridine. So cytosine plus ribose is your cytidine. Okay, it is not cytosine, it is cytidine. Alright, so these are the 3, that is your nucleoside. Alright, yes, clear? Okay, so question number 5. So which of the following is a non-essential amino acid? So I told you to remember Tim, Hall. This is the shortcut. Yes. So you have valine here, you have alanine here, then lysine is also here. Tryptophan and threonine are there. Tyrosine is not your essential amino acid. So 1T stands for threonine. The other stands for tryptophan. Yes. Now give me the answer. No, it is not tryptophan. It is tyrosine only. Oh, which is a non-essential amino acid? Okay, so then I am sorry. So this is not tryptophan, it is threonine and tyrosine, which are essential amino acids. Non-essential is tryptophan. I'm confusing. I'm really sorry. I was right in the first place. So this is for essential amino acid. Okay, so the essential amino acid, yes, valine, lysine is basic, yes. So here it is tryptophan. So now tell me which is a non-essential amino acid. Come on quick. So question number 5. So you say C. So alanine and tyrosine are non-essential. So what does this A stands for? Yes. So valine is an essential amino acid. Lysine is also an essential amino acid. Lysine, leucine, both are essential amino acid. They are asking for non-essential amino acid. So non-essential amino acid. So okay, wait. So threonine, isoleucine, methionine, histidine, adenine, no, alanine, leucine, lysine, phenylalanine, valine, tryptophan. So everything is there. So everything is an essential amino acid, right? C and D, okay, so tyrosine is non-essential. Yes, this is non-essential, right? But alanine is your essential amino acid. It is only D that is essential, right? So give me a minute, let me check. Okay, I found out the mistake. Okay. So yes, those who said option C is absolutely right. It is alanine is non-essential and tyrosine is non-essential. So alanine is one amino acid. It can be synthesized in trace amount by your body as well, and it has to be supplied through diet as well. Yes, C was the right answer. So I guess Prince said C. Absolutely right. So alanine and tyrosine are non-essential amino acids. So this is how you get confused. Even for me it's confusing, then it's definitely confusing for you as well. So questions of these type. Yes, Prince, you're absolutely right. Okay. So question 6. Which of the following is an example of saturated fatty acid? So now only I taught about this. So all your animal fats are saturated fatty acids, right? So what is the answer? So question number 6. Yes, saturated fatty acid. Yes, stearic acid, perfectly right. Okay. A deoxyribose nucleoside is a deoxyribose with a nitrogenous base, sugar and a phosphate. Deoxyribose plus nitrogenous base and a phosphate, ribose plus a nitrogenous base. So what is the answer for question number 7? Sugar and a phosphate is nucleotide. No. So here where is the phosphate? Here they say nucleoside. Yes, so it is deoxyribose, that is a sugar with a nitrogenous base. So this is option A. Question 8. Which of the statement is wrong here? Types of bases in DNA are more than in RNA. In DNA, adenine combines with thymine. In DNA, deoxyribose sugar is found. So uracil is found in RNA. So which is wrong? Yes, Prince. Okay, so which is wrong? So I guess this is the answer for question 7. So give me the answer for question number 8. Yes, so types of bases in DNA and RNA are the same. It is not more. Okay. So question 9, which is wrong about nucleic acid? DNA is single-stranded in some viruses. RNA is double-stranded occasionally. The length of one helix is 45 Armstrong in bDNA. One turn of zDNA has 12 bases. Yes. What is the wrong statement here? Question number 9. Okay, A is for question number 8. Yes, understood. Yes, what is the answer for question number 9? Yes, so the length is, what is the length of the helix? So the length of one helix of your DNA. So the pitch is 3.4 nanometer, right? Or 34 Armstrong. So this is wrong. It's not 45 Armstrong. It is 34 Armstrong or 3.4 nanometer. So nanometer is 10 power minus 9. Armstrong is 10 power minus 10. Yes, question number 10. Primary metabolites play known roles in ecology, chemical process, human welfare, physiological process. Yes, quick. No, it has 10 base pairs per turn, not 20. Not 20, it has 10 base pairs per turn. Chemical process, not physiological process. Yes, Shimpi. It plays a role in physiological process that is within the body. It's not a chemical process, it's a physiological process. In human welfare, no, it's physiological process. Okay? Yes. So it acts within your body. It acts at the level of inside your body, not outside your body. So we say physiological process. Alright. So still this is confusing. So I have to solve a lot of questions from biomolecules. So this alone I understand. So how many of you felt your biomolecule is still tough? Yes. Yes, occasionally you will go wrong. That is fine. But how many of you still feel biomolecule is one chapter I am really feeling tough? Yes, homework question is not there because today I want to teach. After that, I forgot to add homework questions. Okay, so don't worry. So I will give you homework questions in the next class. So you felt easy. That's great. Okay. So Shimpi, okay. So Shimpi says me. I feel tough. So Vishal, okay. So Titan also feels tough. Okay, so I will solve a lot of questions in biomolecules, don't worry. But today I didn't bring any questions because I thought already we have 10 questions. It will take time for me to discuss those 10 questions, okay? You didn't attend previous lecture? Okay, fine. All right, Prince, okay. Anthony felt a bit easy. Okay, so what are biomacromolecules? Okay, so in our fraction, so let me go to the previous slide where I wrote about the fractions. Yes, so we have two fractions, right? So, in this fraction, the weight of these compounds were very less compared to the weight of these compounds. So, these are known as biomacromolecules, or these are simply known as biomolecules because they weigh less than 800 daltons. Okay, tell me this, if it's a living compound, okay, if I have a carbohydrate, if I have a protein, if I have a lipid, how do I weigh? In grams or kg? No, I have a separate term known as daltons. So I measure these compounds, that is biomolecules, in terms of daltons. Okay, yes. Just like in genetics, the map distance is measured in terms of centimorgan. So here, the living components in a living tissue is known as or measured in terms of daltons. So these have less daltons, okay, less than 800 daltons, but this has very heavy molecules. So these are known as biomacromolecules. So these are known as biomacromolecules. Okay, yes. So now going back to that slide. So common to all these compounds is your acid soluble pool, because that contains your organic compounds, right? So the molecules that are present in your acid soluble pool, that is your amino acids, not proteins, only amino acids. Then you have your lipids. Then you have your nitrogenous base. I never said proteins or I never said nucleotides. I am only saying amino acids and nitrogen bases comes in acid soluble pool. Okay, so these have a less weight of 18 to 800 daltons. Fine? Yes. So you have four types which come in your other point, that is your acid insoluble pool. In your acid insoluble pool is where you have protein. If one amino acid can weigh from 18 to 20, a protein is a polymer of amino acid. So this is a biomacromolecule. So is nucleic acid because it contains nucleotide. So in your nucleotide, you have nitrogenous base, sugar and a phosphate group. Then polysaccharides. So this is your carbohydrates. So polysaccharides have individual monosaccharides and to some extent lipids, but this is a myth. I will tell you why. So they come in your acid insoluble portion. So this is known as biomolecules, whereas these are known as biomacromolecules. So what does this signify? So pay attention. So biomolecules, okay, so this biomolecule includes only amino acids, lipids and your nitrogenous bases, and its weight ranges from 18 to 800 daltons. Fine? Okay. So now, yes, so I am coming to the twist that is present. So this is biomolecules or biomicromolecules also you can say. Okay, friends? Now, what is biomacromolecules? So this includes proteins, nucleic acids, polysaccharides and again lipids. So hold on here. So this has more than 800 dalton weight. So this is present in your acid soluble portion. So this is in your acid insoluble portion. Right? Okay. So now pay attention. Ma'am, how come lipids are coming in both the cases? Lipids are here also, lipids are here also. Pay attention. Lipids are not biomacromolecules. They are strictly biomolecules or biomicromolecules. But still, why are they found in the acid insoluble pool as well? So the answer to this question is, your lipid is not present in isolation. It is always conjugated with something like your phospholipids. So which is an integral part of your plasma membrane. So it was not able to be soluble or come out easily with the filtrate because it is conjugated with something else. But as such, your lipids is your biomolecules only. It weighs less than 800 daltons. You got the point? Yes. But why it comes here is because of this reason. It is conjugated as phospholipids, glycolipids. So that is the reason it tends to appear here. But it is strictly a biomolecule. It does not belong here. It has to belong here. Clear? Yes, everyone understood? So now shall we talk about proteins? Yes, everyone? Clear? Yes, Vishal? Any doubt? Yes? Okay, so now talking about your proteins. So proteins are nothing but polypeptides. They are long polymers of amino acid. So this is my protein. So protein has amino acids like this. So polypeptides. So poly means many. So it has many peptide molecules. Clear? Yes. So moving further, so it has linear chains of amino acids linked together by peptide bonds. So this is the linear chain, a straight line, linear, okay. So this has amino acids linked to it. So two amino acids are linked together by a bond known as peptide bond. Okay, fine. So your protein is heteropolymer. So you do not have the same amino acids present. If this is glycine, this is valine, this is phenylalanine, this is methionine. So you have different amino acids which makes up your polypeptide. Thus it is a heteropolymer. You do not have 20 same amino acids making a particular protein. It is always heteropolymer. Okay, so dietary proteins are sources of essential amino acid. So what is the function of your protein? So transport nutrients across the cell membrane. Yes, you have transmembrane protein, you have integral proteins which are present on your plasma membrane. They fight infectious diseases in the form of, how do they fight infectious diseases? In the form of antibodies or immunoglobulins. Right, so these are also nothing but proteins, polymers of amino acid. Some are hormones, some are enzymes, like your insulin, glucagon are hormones, and some are enzymes. Mostly all enzymes are proteins, right? So here some proteins also act as enzymes, like you can say phenylalanine hydroxylase. So this is a protein which acts as an enzyme. So these are the functions of your protein. So collagen is the most abundant animal protein. In animals, where can you find maximum concentration of protein? That is in their tail region. The tail region accumulates a lot of protein. So if you want to isolate collagen, we usually isolate from the tail of the animal. And rubisco is the most abundant protein in case of plants. Why? Because it can bind to both oxygen as well as your carbon dioxide, right? So some important proteins, you have collagen, you have trypsin which acts as an enzyme, insulin which is a hormone, antibody which fights infection. Ribozyme is not a proteinaceous enzyme because that is your RNA which acts as a catalyst. Yes, you're perfectly right, Prince. Then receptor molecules, sensory reception, like your smell, taste. So who takes in, who receives your taste? So that is your gustatory receptor, right? So the receptor and GLUT4. So GLUT4 is your glucose transporter. It helps to transport your carbohydrates or your monosaccharide. So once again, this table is given in your textbook. So please be thorough with this table as well. Okay, so one more thing I want to talk to you about your proteins. So this is the N-terminal end of your proteins or your amino acid. So N-terminal end, and this is the C-terminal end. Why this is called as the N-terminal end? So let me explain. So structure of amino acid. Yes, so this is our amino acid 1. So then I have amino acid 2, amino acid 3, amino acid 4, and then amino acid 5. So how do I know? I have to say this is the first amino acid and not this. So always from this side, so you go from left to right, okay. So since this has an amino terminal, so this portion is known as the N-terminal of the protein or the polypeptide. So this is the N-terminal because here is where you have the amino group, okay. And this is known as the C-terminal of your amino acid because you have the carboxyl group. Clear? Yes, is this clear with everyone? So that is the reason I wrote this is the first amino acid and not this, because the notation or the convention is from left to right. It is from left to right. Clear everyone? Yes? Okay. So moving further, The next one is your polysaccharides. So the acid insoluble pellet also has what is known as polysaccharides or carbohydrates. So your carbohydrates are basically sugars, right? So saccharides also mean sugars. So poly means many. Okay. So polysaccharides are long chains of sugars. So glucose is sugar. So long chains of this is your polysaccharide. It can be glucose or it can be your sucrose also. So that is also polysaccharide. So glucose is a monosaccharide, meaning it cannot be broken down into any further components. This is the basic unit. Okay. But whereas lactose is a disaccharide, it can be broken down into glucose plus galactose. Okay, so this is a disaccharide. So this is a monosaccharide. So group of this forms your cellulose. So this is a polysaccharide because it has many components of your individual monosaccharides. So there are threads, that is monosaccharides as building blocks. So your polysaccharides has monosaccharides as the basic unit. Okay, so cellulose is a polymeric polysaccharide and it has one monosaccharide, that is glucose. So many long chains of glucose makes up your cellulose molecule. So cellulose has repeating structure of only glucose, no other polysaccharide, thus it is known as a homopolymer. So homo is same, right? It has repeating structure of same glucose, so it is homopolymer. Okay. So starch is a variant of your cellulose. It is present or it is a storehouse of energy in your plant cells. You do starch test in your plant using iodine, right? So in animals, the energy is stored in the form of glycogen. Then you have another polymer known as inulin. So inulin is a polymer of fructose. Just like cellulose is a polymer of glucose, so inulin is a polymer of fructose. Okay, so this is your cellulose molecule. So polysaccharide, okay. So if I have glucose molecules like this, right? So glucose is 6 carbon, right? So this is how I write. So 1, 2, 3, 4, 5, 6, right? So this is your glucose molecule. So the right hand side is the reducing end. So what is this? So this is my right hand side. This is the reducing end. So if I want to add glucose molecules, I add only to the right hand side. I do not go and add a glucose molecule here, because this is the non-reducing end. So just like in a polypeptide, that is in your protein, I had N-terminal and C-terminal. In a polysaccharide, I have non-reducing end and I have reducing end. Because only in your reducing end, I can add more monosaccharides. I cannot add a glucose molecule here. I can add only this side. Okay, yes. So if I start from this side to that side, I can only write there. I cannot come back, erase this and write, right? So it's much in the similar way. So the right hand side end is the reducing end and the left hand side is the non-reducing end. So starch forms helical secondary structure. So meaning starch has structure like this. So in this helical structure, it stores iodine. So iodine is produced by your thyroid hormones. So that is why to test the presence of starch, you use iodine, because starch has a helical structure and in between these helices, it can store your iodine. So that is why you use iodine to test starch. Okay. Yes. So this is your glucose molecule. Okay. Yes. So here this is my right hand side end and glucose molecules are added here only. So the structure of how they have drawn and I have drawn is different. So here this is their first carbon, their second carbon, third, 4, 5 and 6. Okay. So here the bond is formed between first carbon of this and the fourth carbon of this glucose. So this is one glucose molecule, this is one glucose molecule. So here, according to their structure, here first carbon, second, third, 4, 5, 6. So the bond is formed between first carbon and 4 carbon. So I write it as 1. So this is alpha linkage. So alpha 1, 4 glycosidic bonds. So you have alpha isomer, conformer and beta conformer, right? So this is alpha. So this is the bond and only in the right hand side I can add, not in the left hand side. Is this clear to everyone? Yes? Done. So this is the structure of starch and it stores iodine inside. So plant cell walls are made up of cellulose. So cellulose is a homopolymer of glucose. Right? So paper which is made from plant pulp and cotton fiber which is also made from plant is cellulosic. So paper is made from two things, cotton pulp and cotton fiber. So they also, the base material is cellulosic. Okay. So you have something known as complex polysaccharide. So what is this complex polysaccharide? They are also building blocks, meaning they are also present in case of your certain proteins. They are amino sugars and they are chemically modified sugars. So they are not normal sugars like your glucose, fructose, but they are chemically modified sugars or they are amino sugars, sugars combined with amino groups. So what are these modified sugars? Glucosamine and acetyl galactosamine. So in certain organisms, yes, so if I have a polysaccharide, so two polysaccharides, so here I have a polysaccharide, but sometimes these two polysaccharides are connected by N-acetyl glucosamine. So this is the function of complex modified sugar and this is a complex polysaccharide. They also connect polysaccharides which are very complex, but this is my modified sugar. It has NAG, N-acetyl galactosamine. So in case of your phylum Orthopoda, okay, exoskeletons have complex polysaccharide known as chitin. So this gives the brown color to your cockroach. Okay, so this is your homopolymer. So this is your homopolymer. So this is your glucosamine. This is your N-acetyl glucosamine. So you have an amino group as well. So this is combined with glucosamine. So this is your chitin. So this gives the brownish appearance to your cockroach. Alright, clear everyone? So with this I will stop. So in the next class we will talk about your structure of DNA. Only that portion is left and then about your enzymes. Okay, yes, what is your doubt in digestion? Yes, Dharini, please ask me. Yes. What is your doubt in digestion? Yes, come on. Epiglottis and glottis. Okay. Okay, so I will explain it on a blackboard. So, what is the passage of food in your alimentary canal? So, you have mouth, then you have buccal cavity, then you have pharynx. So, pharynx opens into your food pipe, that is your esophagus. Then you have your windpipe, that is your trachea. The opening of trachea is called as what? The opening of trachea is known as glottis. Okay, so your food is prevented from entering your windpipe, right? So your epiglottis is okay, so this is a common passage by which your food and air enters inside. So you have to prevent the entry of food inside your trachea, right? So your epiglottis covers your glottis, and that is what is the function of these two. Okay, Dharanji? Yes, clear? Yes, any more doubts? Okay, so please do join me at 4 o'clock tomorrow. I'm going to teach the concept of transport in plants. Okay, yes, fine. So until then, it is me wishing you most and more. So please take care and stay tuned. So this is me signing off. So take care and bye.